So in this chapter, we are going to look at chapter 6, focusing on microbial nutrition and growth. So we're going to start out with chemical requirements for growth. So an essential nutrient is any substance that must be provided to an organism, meaning that the organism needs to take in that substance in order to survive. If we talk about a macronutrient, this is a nutrient that's required in relatively large quantities and plays principal roles in cell structure and metabolism. And so examples of macronutrients would be carbon, hydrogen, and oxygen. Those are needed in all macromolecules, and so we need to take in relatively large quantities of these. On the other hand, micronutrients are present in much smaller amounts and are involved in enzyme function and maintenance of protein structure. These are also known as trace elements, and you might recall back when we talked about chemistry, trace elements are only required in small amounts. And so you're going to see that this is mainly because of their role in enzyme function. Hemoglobin, for example, which is the protein that is involved in transporting oxygen throughout the body, requires iron. Other examples of trace elements would be manganese, zinc, nickel, iodine, etc. They're required in relatively small amounts. So if we look at these different chemical compositions of a cell, so if we look at the organic compounds in the cell, about 50% of the dry weight of organic compounds would be proteins, 20% would be RNA, 3% would be DNA, 10% would be carbohydrates, 10% would be lipids, and then 4% would be miscellaneous. Now, you do not need to memorize this slide. There's no reason for that, so don't worry about that. It's just to give you an idea. In terms of inorganic compounds, right, in terms of dry weight, water would count for none because it's not dry, but other inorganic compounds would make up about 3%. If we look at the elements that are required, notice the big four: carbon, oxygen, nitrogen, and hydrogen. And then we start to get micronutrients, which are required in smaller amounts. So again, like iron, sodium, potassium, etc. And then trace metals would be only 0.3%. So there are different terms for how organisms acquire carbon and how they acquire energy. And so the top two terms are used to describe how an organism takes in carbon. So if we call an organism a heterotroph, hetero means other, troph, feeder. So a heterotroph is an organism that must obtain carbon in an organic form. And you might recall that when we talk about organic molecules contain carbon and hydrogen. So a heterotroph, for example, will take in carbon as sugars, so carbohydrates, proteins, nucleic acids, etc. They have to take in carbon in an organic form. An autotroph, auto, self, troph, feeder. An autotroph is an organism that uses inorganic carbon dioxide as its carbon source. Notice it's inorganic because it does contain carbon, but it doesn't contain carbon and hydrogen. And so autotrophs take in inorganic carbon dioxide as its carbon source. They have the ability to convert carbon dioxide into organic compounds. They are self-feeders. They can make their own organic compounds. These organisms are not nutritionally dependent on other living things. Again, they are self-feeders. There are two terms used to describe how an organism takes in energy. If we call an organism a phototroph, photo refers to sun, troph is eater, this is going to be a microbe or an organism that photosynthesizes, that uses sun for energy. If we call an organism a chemotroph, this is an organism that gets energy from chemical compounds. And so the top two, again, are describing how an organism gets its carbon source. The bottom two terms are describing how an organism takes in energy. So this slide is just breaking down how organisms can be classified. So we'll start with our autotrophs. Remember that our autotrophs are going to be our self-feeders. Notice for this, their carbon source is always carbon dioxide, but they have different names depending on their energy source. So if we call an organism a photoautotroph, photo refers to the fact that they take in energy and sunlight. Auto refers to the fact that they take the carbon as carbon dioxide. And example organisms of photoautotrophs would be photosynthetic organisms such as algae, plants, and cyanobacteria, which is a type of bacteria that does photosynthesis. We can have what are called chemoautotrophs. Chemoautotrophs are going to use chemicals. And so some of them can be referred to as chemoorganic autotrophs. Chemoorganic means that they take in energy as organic compounds. They use chemicals, but organic compounds is the chemicals. But they will utilize carbon dioxide. Notice the autotroph. They will utilize carbon dioxide as their carbon source. And an example of this would be our methanogens, our methane producers. We can have what are called chemolithoautotrophs. So chemicals for energy. The litho is referring to the inorganic compounds. So their energy source is inorganic compounds or minerals. Because they're an autotroph, they take in carbon as carbon dioxide. And an example of this would be thiobacillus, or what we call rock-eating bacteria. They will utilize inorganic compounds. Next, we get to our heterotrophs. And we can have heterotrophs, remember, take in carbon in organic forms. So notice for all of these, their carbon source is organic. If we have a photoheterotroph, photo refers to energy in light, so sunlight. Again, hetero refers to organic carbon sources. And so examples of this would be purple and green photosynthetic bacteria. Chemoheterotroph, right? A chemoheterotroph is going to use chemicals for energy. So the metabolic conversion of nutrients from other organisms. They will utilize organic compounds for carbon source. And so organisms that would fall in this category would be protozoans, fungi, many bacteria, animals like ourselves, etc. So this is a very big group of organisms, right? We are chemoheterotrophs. We have to consume our food in order to survive. We are not self-feeders. Chemoheterotroph, there is a class of that which is referred to as a saprobe. And a saprobe is going to get energy by metabolizing the organic matter of dead organisms. So these are organisms that feed on dead, cast-off materials that other organisms typically don't want. They are a heterotroph, so they do take in carbon as an organic compound. And so examples of a saprobe would be fungi, bacteria, basically your decomposers in an ecosystem. If we talk about a parasite is a type of chemoheterotroph, but it utilizes the tissues or fluids of a live host. They will take in organic carbon sources. And examples of this would be various parasites and pathogens. Those could be bacteria, those could be fungi, they could be protozoans, they could be animals. There's a wide variety of different parasites, and in lab especially, we will talk about different types of parasites. So, parasites derive nutrients from the cells or tissues of a living host. A pathogen is going to cause damage to tissues or even death. And these organisms that are parasites can range from viruses to helminths, helminths being animals. Remember, those are our parasitic worms. We can't talk about parasites based on where they're found. So if we refer to something as an ectoparasite, ecto is like outside. These are organisms, these are parasites that live on the body. They're not in the body, but they're on the body. We can talk about endoparasites. Endo means within. They live within the body, in the organs and the tissues. We can have intracellular parasites. These are parasites that live within cells, so they must replicate within a host cell. An obligate parasite is one that's unable to grow outside of a living host. So examples of this would be leprosy bacillus, syphilis, which is a spirochete. Viruses would be an obligate intracellular parasite, right? So they're not only can they grow inside of a host, but they can only reproduce within the cells of a host. So viruses would be an obligate intracellular parasite. They only reproduce within the cells of a host. So now we're going to look at what are the chemical requirements for growth. What chemicals do bacteria need in order to grow? So the first and the most abundant is going to be carbon. And so carbon acts, its function is that it functions as a structural backbone for organic molecules, right? So our proteins, our nucleic acids, our carbohydrates, these are all going to contain carbon. Carbon is also used as an energy source. Now, what do organisms use carbon for? They use carbon to make all macromolecules. So organisms must have a carbon source in order to make all of their macromolecules. How they obtain carbon is different depending on their nutritional classification. So again, if we call an organism a heterotroph, the way that they take in carbon is through organic carbon sources. If we call an organism an autotroph, they will take in carbon dioxide as their carbon source. So these are the ways that organisms can take in their carbon, either through organic compounds or through carbon dioxide. So this slide is just kind of breaking down different examples of nutritional classifications. So again, if we start with chemotrophs, chemotrophs are going to use chemicals for energy. We can then break those down further by their carbon source. So if they use organic compounds, again, we're going to call these a chemoheterotroph. Chemoheterotroph, right? They use chemicals for energy, organic compounds for carbon. We can then further divide these by their final electron acceptor. And what that means is that when we talk about metabolism and how organisms obtain energy, you're going to learn that they have different types of final electron acceptors, meaning when we break down the food, the electrons have to go somewhere. So if the final electron acceptor is oxygen, this would include animals, for example, right? We have to breathe in oxygen, and that's because we need oxygen to be the final electron acceptor. Most fungi, protozoans, bacteria, etc. Those would be chemoheterotrophs that use oxygen as their final electron acceptor. Now, some are going to use final electron acceptors that are not oxygen. Those can then be further divided by, do they use organic compounds as their final electron acceptor? If they do, these are organisms that do fermentation. So when we talk about metabolism, again, we're going to talk about fermentation, and organisms might use pyruvate as the final electron acceptor. That's used for lactic acid fermentation. Others might use acetaldehyde, which is used for alcoholic fermentation. And so an example organism that is fermentative would be Streptococcus. This would be a type of bacteria. Think of Streptococcus pneumoniae or Streptococcus pyogenes, which causes strep throat. Those are organisms that do fermentation. That's how they get their energy. Some will use an inorganic compound as the final electron acceptor, but that final electron acceptor is not oxygen. These organisms will still use an electron transport chain, and an example of this would be Clostridium. So organisms in this category might use something like nitrate, NO3-. They might use sulfate, SO4 2-. They might use carbonate, etc. Those are all inorganic compounds. They still use an electron transport chain, but that final electron acceptor is not oxygen. And so again, you're going to learn more about this when we talk about metabolism. If we now go over here, carbon source is now carbon dioxide. We call these chemoautotrophs. Remember, auto refers to self-feeders. They don't take in organic carbon, they take in inorganic carbon. And so examples of this would be hydrogen, sulfur, iron, nitrogen, and carbon monoxide oxidizing bacteria. So these do metabolism just a little bit different. Now, if we look at our phototrophs, remember that a phototroph is going to get energy from sunlight. We can then further break those down based on their carbon source. If they use organic compounds, those are going to be our photoheterotrophs. Again, photo, sunlight, hetero, organic compounds, troph, feeder. So examples of this would be our green non-sulfur bacteria and purple non-sulfur bacteria. If the carbon source is carbon dioxide, this would be photoautotrophs. Again, photo using sunlight, auto means using carbon dioxide, troph, feeder. We can then break those down further by, do they use water to reduce carbon dioxide? Meaning, is this photosynthesis going to use water? So if the answer is yes, that is an organism that would do what's called oxygenic photosynthesis, meaning that they will produce oxygen as a product. So plants would be an example of this, algae would be an example of this, cyanobacteria would be an example of this, etc. But not all of them will use water, and they won't always produce oxygen. And so those are referred to as anoxygenic, meaning in the absence of oxygen. They do not produce oxygen in their process. So anoxygenic photosynthetic bacteria, this would be our green and purple bacteria, for example. And so these are just some examples of different types of nutritional needs that an organism might fall into. So, question for you. Which of the following terms describes an organism that derives its energy and carbon from organic molecules? Would that be red, a photoautotroph, yellow, a chemoheterotroph, green, a lithoautotroph, blue, chemoautotroph, or purple, photoheterotroph? So take a minute, pause your video. When you are ready, go ahead and push play to hear the answer. So if you said yellow, a chemoheterotroph, you would be correct. Because again, chemo refers to chemicals. Notice I said that it takes in energy from organic molecules. That's energy from chemicals. Hetero refers to how they take in carbon. And notice I said that their carbon is from organic molecules. So this would be a chemoheterotroph. Photoautotroph, again, photo would be one that uses sunlight for energy. Auto referring to carbon dioxide for their carbon source. So that's not right. A lithoautotroph, it's also the same as a chemolithoautotroph. They use chemicals for energy, but litho refers to inorganic chemicals. Auto is referring to using carbon dioxide, not organic molecules. A chemoautotroph is just more broad. They use chemicals and they use carbon dioxide. Photoheterotroph, photo is going to use sunlight for energy and organic materials, organic sources for carbon. And so the one that's going to use energy and carbon from organic molecules would be our chemoheterotroph. So the next chemical that we're going to talk about in terms of requirements that bacteria need, hydrogen. Hydrogen is used to make all macromolecules. So just like carbon, hydrogen is also found in all macromolecules. And that's because, right, they're all organic compounds. Organic compounds are going to have carbon and hydrogen. How do organisms acquire hydrogen? Well, they take in hydrogen through organic compounds, right? So taking in glucose, for example, or proteins, etc. And they're acquired through several inorganic compounds as well. Water, for example, would be a way for a cell to take in hydrogen. Salts, for example, and then certain naturally occurring gases like hydrogen sulfide, methane, and hydrogen gas. These would be another way that organisms could potentially take in hydrogen. What is the role of hydrogen in the cell? Well, hydrogen helps cells to maintain their pH, because remember that pH is a measurement of hydrogen ion concentration, is useful for forming hydrogen bonds between molecules. So like that's what gives water its very important properties because of its ability to form hydrogen bonds, right? It demonstrates cohesion, ice is less dense than water, etc. That's all due to hydrogen bonding. Hydrogen also serves as a source of free energy in respiration. What you're going to see is that it's actually a hydrogen ion gradient that is what is going to allow the cell to make the bulk of the ATP. So hydrogen ions can also serve as a source of free energy. Next, we have our nitrogen. Nitrogen is used to make which macromolecules? So in lab, this has been discussed several times. For nitrogen, it's used in proteins and nucleic acids. Nucleic acids include DNA, RNA, and even ATP. They all require nitrogen. So proteins have amino acids as their building blocks, and the amino acids has an amino group which contains nitrogen. Nucleic acids, the building blocks of nucleic acids are called nucleotides. Nucleotides have three parts: a sugar, a phosphate, and a nitrogenous base. Nitrogenous base tells you that the base has nitrogen. So cells need nitrogen because they need it to make their proteins and their nucleic acid. That's what it's used to make. How do organisms take in nitrogen? Well, most bacteria decompose proteins to get a source of nitrogen. So the most common way that organisms take in nitrogen is by degradation of proteins, because the proteins are going to have nitrogen in it. However, some bacteria use ammonium, so NH4 plus, or nitrate, NO3 minus. And so they might take in their nitrogen in that way, so by one of these other compounds. There are a few bacteria that will use nitrogen gas from the atmosphere. This is rare. This is why bacteria play such an important role in what we call the nitrogen cycle, meaning how nitrogen moves through an ecosystem. Because bacteria are essentially the only organisms that are able to utilize atmospheric nitrogen. Atmospheric nitrogen makes up about 70% of the gas in our atmosphere. So it's a very abundant gas, yet most organisms can't take in nitrogen in that form. When they do this is referred to as nitrogen fixation. So an example of this would be photosynthesizing cyanobacteria. They're able to do nitrogen fixation. They're able to utilize nitrogen gas. Again, this is unique. This is unique to bacteria. Next, we have sulfur. Sulfur is used in amino acids, so to make proteins, because we have cysteine, for example, which is an amino acid that contains sulfur. It's also found or used to make vitamins. Vitamins are basically organic compounds that are used to help enzymes function. And sulfur can play a role, and it can be found in these vitamins. How do organisms take in sulfur? Well, most bacteria decompose proteins for their sulfur source. So that is the primary way that most bacteria take in sulfur. However, some bacteria will use sulfate ions. Some bacteria will use hydrogen sulfide, H2S. This is what's discussed in our TSI test, for example. So this is the way that organisms can take in sulfur, and then the organism uses that sulfur to make amino acids, so to make proteins, and to make vitamins. In the case of phosphorus, phosphorus is used in nucleic acids, DNA, RNA, ATP, because again, phosphorus is going to be found in our nucleic acids because our building blocks of nucleic acids are called nucleotides. Again, nucleotides have three parts: a sugar, a phosphate, there's the phosphorus, and the nitrogenous base. So organisms have to take in phosphorus to use it for synthesis of nucleic acids. Phosphorus is also found or also used to make cell membranes because cell membranes are made of a phospholipid bilayer. Those contain phosphorus in them. A major source of phosphorus for organisms would be phosphate ions, so PO4 3-. That's a source of phosphorus for organisms. And that phosphate ion can be found in rocks as well as oceanic mineral deposits. And so that is a major, what we call abiotic reservoir, meaning it's a main place where phosphorus is stored outside of living things. It's found in rocks and oceanic mineral deposits. So we already talked about carbon, hydrogen, nitrogen, sulfur, and phosphorus. And next we're going to come to our trace elements. And our trace elements are inorganic elements that are required in small amounts. And remember that inorganic elements are going to lack carbon and hydrogen. So they lack carbon and hydrogen. And so again, they're required in small amounts. So this would be like our metal ions, for example, iron, copper, zinc. And these trace elements are usually going to act as enzyme cofactors, meaning that they help make enzymes function. And so we talked about in our chemistry chapter about how iron is needed in hemoglobin. And hemoglobin, remember, is the protein in our red blood cells that's responsible for picking up oxygen and carrying oxygen to all the tissues in our body. And we need iron in order for hemoglobin to function properly. Iron is a trace element. We need it in small amounts, and it helps hemoglobin, that protein, to function. So next we have oxygen. And if you think back to our macromolecules, you might recall that oxygen is used in all macromolecules, meaning that organisms need to have a source of oxygen in order to make all of their macromolecules. They need it for their carbohydrates, they need it for their proteins, they need it for their lipids, they need it for their nucleic acids. All of our macromolecules requires oxygen. Now, how is that oxygen obtained? Well, typically it's going to be obtained through organic compounds. So again, our macromolecules like our sugars or our proteins, etc. It's also obtained through inorganic salts such as sulfates, phosphates, nitrates, and even water. So these are sources of oxygen. Oxygen plays an important role in the structural and enzymatic function of the cell. Now, in terms of gases, there are two main gases that have an effect on microbial growth. And those two atmospheric gases that affect microbial growth are going to be oxygen, O2 gas, and CO2, which is carbon dioxide. O2 has the greatest impact on microbial growth, meaning that in some cases, some bacteria can grow in the presence of oxygen, others can only grow in the absence of oxygen. And so O2 plays a big role in microbial growth. O2 also is an important respiratory gas. So in aerobic cellular respiration, O2 acts as the final electron acceptor during cellular respiration. And it's also a powerful oxidizing agent. So microbes fall into one of three categories depending on its oxygen need. So the first category would be those that can use oxygen and can detoxify it. And what you're going to see is that when organisms use oxygen, they produce these toxic reactive oxygen species, meaning these forms of oxygen that are unstable. And so bacteria that use oxygen need to have enzymes in order to detoxify those toxic forms of oxygen. We can also have some bacteria that can neither use oxygen nor detoxify it. So for these bacteria, O2 is going to be toxic to them. So they don't use it and they don't use it because for them, they're not able to detoxify it. And then lastly, we have those that do not use oxygen, but can detoxify it. So what that means is they can grow in the presence of oxygen. They grow where O2 is found, and therefore they're able to detoxify the toxic reactive oxygen species, but they still do not use oxygen. And so we'll talk about some examples of these in just a minute. Now, as oxygen enters into cellular reactions, it's transformed into several toxic products. So again, when O2 acts as that final electron acceptor, it can form these toxic reactive oxygen species. And so the first form is going to be our singlet oxygen. This is an extremely reactive molecule, and it can damage and destroy a cell by the oxidation of membrane lipids. So this is not stable in the cell. We have our superoxide ions, so our O2 minus. Again, these are highly reactive. We have hydrogen peroxide, H2O2. It's toxic to cells, and it's also used as a disinfectant. Probably at some point, you've probably had hydrogen peroxide poured on a wound, and you've seen that bubbling, and later on, you'll see why. And then we have our hydroxyl radicals, so our OH minus, which are also highly reactive. And so these forms of oxygen are toxic because they steal electrons from neighboring atoms. And so because of the fact that they're going to steal electrons from neighboring atoms, that makes these forms of oxygen highly unstable. So what that means is that organisms that grow where oxygen is found have developed enzymes that scavenge and neutralize reactive oxygen byproducts. And so this is going to be a two-step process. And this two-step process requires two enzymes. The first enzyme is called superoxide dismutase, which you will see abbreviated SOD. And this enzyme is used to neutralize superoxide ions. So they will neutralize those superoxide ions, and they will convert those superoxide ions into our products. And we have two products in this case. We have hydrogen peroxide, and we have O2 gas. Now, remember I said that hydrogen peroxide is also unstable. So there's going to be a second enzyme needed to neutralize the hydrogen peroxide. But first, let's talk about what types of organisms would have SOD. And so this is used by organisms that grow in the presence of O2. So whether or not the organism uses oxygen doesn't really matter. Organisms that grow in the presence of oxygen need to have that superoxide dismutase. They need to be able to neutralize those superoxide ions that can form. Now, when superoxide ions are neutralized and they produce hydrogen peroxide, we need a second enzyme to neutralize the hydrogen peroxide. And so that second enzyme is one that we talked about in lab, and that enzyme is catalase. And so this is used to neutralize hydrogen peroxide. So catalase is an enzyme that's used to neutralize hydrogen peroxide. So it's going to take that hydrogen peroxide and it's going to neutralize it, and the products are going to be water NO2 gas. So notice that in this case, we form more O2 gas. So for catalase, it's not just used by organisms that grow in the presence of oxygen. In this case, it's actually used by organisms that use O2 as their final electron acceptor. So not only do they grow in the presence of oxygen, but they actually use O2 as their final electron acceptor. Because if the organism does not use O2, they're not going to want to use catalase and produce more oxygen. Notice that this product is going to produce more O2. That doesn't want to happen if the organism is not going to utilize that O2. So some organisms have a third enzyme instead of catalase. They have an enzyme called peroxidase. And so for peroxidase, we still have hydrogen peroxide being neutralized, but our products in this case are going to be simply water. It's not going to form more O2 gas. Now, I didn't balance that equation. I'm not worried about you knowing that, but I want you to know what the purpose of those enzymes is. So peroxidase is used to neutralize hydrogen peroxide, and it's going to produce water in the process. It's not going to produce more oxygen. So this is used by organisms that grow in O2, but don't use it. And again, that's because if the organism does not use O2, it doesn't want to form more O2 gas. So instead of using catalase, instead the bacteria would use peroxidase. They would still neutralize hydrogen peroxide, but they're going to form the product of just water. And so again, it's the presence of that catalase that's going to cause the bubbles when you pour hydrogen peroxide in a wound. The hydrogen peroxide that causes the bubbles is not only from bacteria that might have gotten into the wound, but your cells also contain catalase. And when you get a cut and those cells are damaged, those cells will also release catalase, which would also then produce the oxygen bubbles when you pour hydrogen peroxide in that wound. And so these are the enzymes that bacteria need to have if they're going to grow where O2 is found. They have to have these enzymes to be able to neutralize those toxic oxygen species. So we have different terms for an organism's oxygen requirements. If we call the organism an aerobe, what that means is that the organism uses molecular oxygen, so O2, as the final electron acceptor. So as our final electron acceptor in aerobic respiration. And so within the umbrella of aerobes, we can have organisms that are obligate aerobes. And just like the name suggests, they are obligated or they require O2. And an example of this would be Pseudomonas aeruginosa. And Pseudomonas aeruginosa causes infections in burn victims and patients with cystic fibrosis, which we will abbreviate CF. So Pseudomonas aeruginosa causes infections in burn victims, and in fact, it's the leading cause of death in burn victims. And you might recall that Pseudomonas also produces a pigment called pyocyanin. Pyo again refers to pus, cyan refers to blue, which means that when this organism is growing in a wound, it causes the wound to appear to have blue pus. And so that is a hallmark of a Pseudomonas aeruginosa infection. It also is very problematic in patients with cystic fibrosis. Cystic fibrosis, you may recall, is a genetic condition that people are born with. And in cystic fibrosis, patients have defective chloride channels. Because they don't move chloride properly, they also have problems with water imbalance, and they end up with these secretions in the lungs. And Pseudomonas really likes water. And so if Pseudomonas happens to get in these secretions in the lungs of these patients, it causes a major problem because they can't clear it out of their lungs. And so Pseudomonas aeruginosa would be an example of an obligate aerobe. Other examples of obligate aerobes would include most fungi, protozoans, and many other bacteria such as Bacillus species. So you might recall that Bacillus is a bacterium that produces endospores, and it is an aerobic species, meaning that it grows where O2 is required. And also under obligate aerobe, we have Mycobacterium tuberculosis, which causes lung TB, tuberculosis. Next, we have our microaerophile. Micro refers to little, aero refers to air, phile is lever. So these are our little air levers. These require low O2 concentrations. And so an example of this would be Helicobacter pylori. And you might recall that Helicobacter pylori is the infectious cause of ulcers. And so what happens is that if Helicobacter pylori, if this bacteria happens to get into the stomach, remember that an advantage that Helicobacter pylori has is that it produces urease enzyme. And what that does essentially for this bacteria is that if this bacteria gets into the stomach, in the stomach is the substrate for the enzyme, urea. And so if Helicobacter pylori gets into the stomach, it's going to take that urea in the stomach and it's going to hydrolyze it. It's going to break it down. And one of the products that gets produced during this is going to be ammonia. And the advantage for the bacteria to produce ammonia is that the stomach is a very acidic environment. Most bacteria don't survive well at that acidic of a pH. So because Helicobacter pylori produces urease, it's able to produce ammonia. It's able to neutralize the stomach acid and therefore grow in the lining of the stomach. And it erodes and it damages the lining of the stomach, which then leads to ulcers. And so this bacteria is the infectious cause of ulcers. And again, remember that not all ulcers are caused by bacteria. Other examples of organisms that are microaerophiles would be organisms that live in soil, in water, or in mammalian hosts in places that are not directly exposed to the atmosphere. So another example of a bacteria that would be a microaerophile would be something like Neisseria gonorrhoeae. Neisseria gonorrhoeae, remember, is the cause of gonorrhea. Gonorrhea is a sexually transmitted infection. And this bacteria is a microaerophile. It needs a low concentration of oxygen. And so this bacteria is going to be found on the opening of the urethra. So it's not in full contact with oxygen. It's not fully anaerobic, but it has a very low concentration. And so that would be an example of a microaerophile. If we call an organism an anaerobe, what that means is one of two things. Either the bacteria does not require O2, so in some cases they use O2 if it's available, but they can grow in the absence of O2. In some cases that means that they don't use O2 at all, or it could be that O2 is actually toxic to them. They're actually missing those enzymes that we talked about in the last slide, which allows the organism to be able to detoxify those reactive oxygen species. And so we're going to look at some different examples of some anaerobes. So the first one that we're going to talk about is going to be the obligate anaerobe. And for our obligate anaerobe, it's going to cease growth or die in O2. So cease growth or die in O2. Notice I did not just write that it causes them to die. And the reason for that is that an example of an obligate anaerobe in many cases is the genus Clostridium. So many Clostridium would fall under being an obligate anaerobe. Now, if you recall, Clostridium produces a structure when conditions become harsh or unfavorable. Clostridium would produce the structure to protect themselves. Now, in the case of Clostridium, that condition could be that oxygen is present. So if O2 is present and O2 is toxic to them, they would produce the structure that would protect them until conditions became favorable again. And so think back to what we've learned and think about what structure Clostridium produces that allows them to not die from O2, but instead would allow them to cease growth. And so what structure would that be? And so if you were thinking endospores, you are correct, right? Because endospores are a structure made of keratin that bacteria can produce when conditions become harsh. In the case of Clostridium, because O2 is toxic to them, in the presence of O2, they would form this endospore to wait it out until conditions became favorable again. And so that's why I was careful to write cease growth or die in O2, because in some cases it's not necessarily going to kill them. However, it will at least inhibit their growth. So we're going to put example, Clostridium perfringens. And Clostridium perfringens causes gas gangrene. And so you might recall that we've talked about this organism, and that is that if Clostridium perfringens happens to get into a wound, it can start to cause necrosis of the tissue. The tissue starts to die. And as a result, when the tissue dies, that makes the environment anaerobic. Because if the tissue is dead, it's no longer getting blood flow. And if it's not getting blood flow, it's not getting enough oxygen. And that further helps Clostridium to be able to grow. And so what happens is that tissue that's necrotic, that has died, the bacteria is going to start to grow and to spread, and that tissue is going to continue to die. And remember that we talked about that oftentimes, because it's difficult to get the antibiotic to the site of the infection, one of the more common things that happens in response to gangrene is that oftentimes the treatment involves amputation of the limb. So if it was the finger, it might be the finger. If it was the whole hand, it could be the hand. But basically that the treatment might include amputation because it's difficult to treat gangrene. Now, there have been some examples of using hyperbaric chambers. So basically to put the patient in a high oxygen environment, which remember that Clostridium does not grow well in oxygen. It inhibits their growth. And so this would help to hopefully treat Clostridium. However, in severe cases, sometimes that's not enough. The hyperbaric chamber is not enough, and they still end up having to do an amputation anyways. And so this would be an example of an obligate anaerobe. Other bacteria that would be obligate anaerobes include often intestinal bacteria. So bacteria that live in the intestinal tract would often be an obligate anaerobe. For them, oxygen is toxic. The next category of organisms that we have would be our facultative anaerobes. And our facultative anaerobes grow with or without O2, but grow better. They grow better with O2, meaning that they will use it when given the choice. So for these organisms, they do use oxygen. However, they don't require it. And so that's why I wrote grows with or without oxygen, but grow better with. Now, the reason that they grow better with oxygen is that if the organism were to do aerobic respiration using O2 as the final electron acceptor, if they use oxygen as the final electron acceptor, they can get 30 to 32 ATP per glucose. On the flip side, if the organism grows anaerobically and they use fermentation, they only get 2 ATP per glucose. So notice that using aerobic respiration gives you a much higher ATP yield than fermentation does. And so if the organism is a facultative anaerobe, right, they can grow with or without, but if given the choice, they're going to grow with O2 and use O2 because for them, using oxygen gives them the highest ATP yield. And so an example of this that you've seen this would be when we did our standard plate count. So remember that our standard plate count, the purpose of our standard plate count was to isolate individual colonies of bacteria and determine the titer, so the concentration of bacteria in the solution. And so when we did this, we took our E. coli and we mixed it with the agar and then poured our plates. And so you might recall that we had some colonies on the top of the agar and some colonies embedded in the agar. And if you recall what that looked like, you might recall that the colonies on the top of the agar were very large, and the colonies that were embedded in the agar, that were in the middle of the agar, those colonies were teeny tiny little dots. And again, back then I told you that the reason for that is that if O2 is available, so the top of the agar, bacteria is going to get a lot more ATP per glucose than the bacteria that's growing anaerobically within the agar. And so that allowed you to see that in fact facultative anaerobes grow better with O2. So an example of this, E. coli. So Escherichia coli. Okay, you can write out Escherichia. You've seen it many times. So example is E. coli. And E. coli, for example, is the cause of urinary tract infections. About 90% of all urinary tract infections, what we call UTIs, are caused by E. coli. And so E. coli would be an example of a facultative anaerobe. In lab, we also saw another example of this, like Staph aureus, for example. Staph aureus is also a facultative anaerobe. And many gram-negative intestinal bacteria would also fall under being a facultative anaerobe. So those would be examples of facultative anaerobes. And the last one is going to be our aerotolerant anaerobes. Aerotolerant means that they tolerate O2, but they don't use it. So we're going to write can grow in O2, but don't use it. So we can put can grow with or without oxygen. So notice there's a key distinction between a facultative anaerobe and an aerotolerant. Both of those two categories can grow with or without oxygen. The difference between them, however, is that the facultative anaerobe will use O2 if O2 is present. An aerotolerant anaerobe can grow with or without, but even if O2 is present, it's not going to use it. It's not going to use it as its final electron acceptors. For example, examples of aerotolerant anaerobes would be Streptococcus, Pyogenes, which causes strep throat, and these organisms are strict fermenters. So for strep pyogenes, even if O2 is present, it's not going to matter. It's not going to use O2 even if O2 is present. Other types of bacteria that would fall under being aerotolerant anaerobes, some lactobacillus, for example, other streptococcus species, etc. So those would be examples of aerotolerant. They grow with or without O2, but they don't use it. And so again, you want to be careful. That's the distinction between the facultative anaerobes and the aerotolerant, is that the facultative anaerobes can grow in both conditions, but if O2 is present, they will use it. Aerotolerant is not going to use it. So now let's talk about why organisms are one of these categories of organisms. And so you might recall that in lab, we talked about our thioglycolate broths. And that our thioglycolate broths use a reducing media to create an anaerobic environment. And so in lab, we talked about what was in thioglycolate broths, and you might recall that there are two chemicals that are in the thioglycolate broth, which helps to make the environment anaerobic. And so if you recall back, we talked about L-cysteine, which is a dimer of cysteine amino acids. So we had L-cysteine and we had our sodium thioglycolate. Those were our ingredients that both were used to reduce O2 to water, which makes the bottom of that tube anaerobic. You might also recall that in this media, we have an O2 indicator, and that O2 indicator was rizagirin. And rizagirin is pink when O2 is present. And so we talked about what our thio broths look like for each type of organism. So again, if it's an obligate aerobe, it's going to grow only in the top, only where the rizagirin is pink. In the facultative anaerobe, it's going to grow best where O2 is present, so best where it's pink, but it will still grow in the bottom part. It'll still grow in the anaerobic part. In the case of an obligate anaerobe, it's not going to grow where the tube is pink. It's going to grow towards the bottom. It might be settled at the very bottom. It might be all throughout the part underneath the pink. But the idea here being that it's not going to grow where it's pink, because pink is where the O2 is present. Next, we have our aerotolerant. Aerotolerant, remember, means that they tolerate O2, but they don't use it. So what you're going to see in this example is that it might grow evenly throughout. It grows equally well in the top as it does in the bottom. And then lastly, we have our microaerophiles, and our little air lovers are going to grow right on the junction between where it's pink and it's not pink. And so in lab, we saw it growing right underneath where the pink is. So now what I want you to think about is, for each of these types of O2 requirements, I want you to think about whether or not they would have superoxide dismutase. So meaning give it a plus if they produce it or a minus if they don't. And same thing with catalase. Does the organism produce catalase? If it does, give it a plus. If it does not, give it a minus. And so take a minute, and I want you to attempt this on your own before hearing the answer. So if you need to, pause your video, and when you're ready, turn it back on, and I'll go over the answer with you. Okay, so hopefully you tried this on your own, but let's review. So superoxide dismutase, remember, is used to neutralize superoxide ions, so superoxide radicals. So for our superoxide dismutase, remember that it's produced by organisms that grow in O2. So whether or not they use O2 does not matter. If they grow where oxygen is present, they will produce superoxide dismutase. So obligate aerobes, do they produce superoxide dismutase? And the answer is yes, right? They grow where oxygen is found, therefore they must have SOD. Facultative anaerobes grow with and without because they grow where O2 is present. They will have their superoxide dismutase. Obligate anaerobes do not grow where O2 is found. They do not have superoxide dismutase. And so in this case, the organism is an obligate anaerobe. What makes them an obligate anaerobe is the fact that they don't produce this enzyme. They don't have this gene. They don't produce this enzyme. And as a result, because they can't neutralize superoxide ions, those superoxide ions would be toxic to them and would kill them. And that is why they are an obligate anaerobe, because they do not produce that superoxide dismutase. So that would be negative. Aerotolerant anaerobes do grow where O2 is found. That would be positive. And then lastly, we have our microaerophile, our little air lovers. They do grow where O2 is found, but only where we have a low concentration. And that's because they do produce SOD, but only a little bit. So they have just a little bit of SOD present, which is why they can grow where O2 is present, and they actually even use O2. But they can't grow where oxygen concentrations are high because too much of those reactive oxygen species would be toxic. They don't produce enough enzymes to be able to neutralize the full amount of oxygen in the atmosphere. Next, when we look at catalase, catalase is not produced by organisms that grow in O2, but instead is produced by organisms that use O2. So that's our distinction. So now when we're talking about catalase, catalase is neutralizing hydrogen peroxide, and it's produced by organisms that use O2 as their final electron acceptor, not just grow in the presence, but actually use it. So if we look at our obligate aerobes, would they produce catalase? The answer is yes, because they use O2. Facultative anaerobes, they still use O2 as their final electron acceptor. Therefore, they would produce catalase as well. Obligate anaerobes, right? They don't use O2. They are catalase negative. So again, the reason that obligate anaerobes are obligate anaerobes is because they're negative for SOD and catalase. They don't produce either of those enzymes, and therefore O2 is toxic. Now, when we get to our aerotolerant anaerobes, remember that those grow where O2 is found, but they don't use it. So this organism is going to be negative for catalase, but remember it's going to have peroxidase instead. And you might recall that peroxidase is used to neutralize hydrogen peroxide, but it doesn't produce more O2 as a result. It just produces water. So aerotolerant anaerobes will not produce catalase because they don't use O2. And then our microaerophiles, these would produce a little bit of catalase. And again, that's what allows them to live where some O2 is present, but only in low concentrations. So, question for you. An organism that has peroxidase and superoxide dismutase, but lacks catalase, is most likely is it red obligate aerobe, yellow aerotolerant anaerobe, or green obligate anaerobe? So go ahead and think about this for a moment. And when you're ready, turn it back on, unpause your video to turn it back on and listen to the answer. If you said aerotolerant anaerobe, you would be correct because this is an organism that can grow with or without O2, but doesn't use it. And so because they don't use it, they would still produce SOD, they would still produce the superoxide dismutase, but they don't produce catalase. Instead, they produce peroxidase because peroxidase can neutralize hydrogen peroxide without producing more O2. An obligate aerobe, because they use O2, they would have catalase and not peroxidase. Obligate anaerobe would have none of those enzymes. It wouldn't have superoxide dismutase, it wouldn't have peroxidase, it wouldn't have catalase. And it is an obligate anaerobe because it lacks those enzymes. So, in addition to oxygen concentrations playing an important role in microbial growth, carbon dioxide also plays an important role. And so if we call an organism a capnophile is an organism that grows best at higher CO2 tensions than is normally present in the atmosphere. So these organisms would require typically a lower oxygen concentration, but a higher CO2 concentration. And the way that we would grow organisms that are capnophiles can be grown in a candle jar. And so what that means is that if you recall in lab, we used our anaerobic jars, and in the anaerobic jars, we put those anaergen chemicals, and those anaergen chemicals, their job was to reduce O2 to water, and that's what made the jar anaerobic. In the case of a capnophile, we don't need to get rid of all O2, but we need to have less O2 and more CO2. And that can be achieved by putting a candle in that jar. The candle is going to utilize some of the oxygen and it's going to produce some CO2. And so it's going to create the ideal environment to be able to culture a capnophile. These are important in the initial isolation of certain organisms from clinical specimens. For example, Neisseria would be an example of a capnophile that can cause gonorrhea, meningitis, brucella, which causes an undulate fever, strep pneumoniae, which causes pneumonia. These would all be examples of capnophiles. So now we're going to look at what are the physical requirements for growth. So we looked at the chemical requirements, right? So which elements bacteria need to grow, but now we're going to look at what are their physical environmental conditions. So we're going to start with our temperature first. And so we have different terms when we talk about an organism's temperature requirements. And the first one is the minimum temperature. And this is the lowest temp that permits a microbe's continued growth and metabolism. And below this temperature, activity stops. Meaning that once you get below the minimum growth temperature, it doesn't necessarily kill the bacteria, but it will inhibit their growth. They won't be able to grow at these low temperatures. The maximum growth temperature, on the other hand, is the highest temperature at which growth and metabolism can proceed before proteins are denatured. So for the maximum growth temperature, if the temperature goes above that maximum, not only will it inhibit their growth, but in many cases it will kill the bacteria. Because at those high temperatures, the proteins denature, right? Which means, again, that the proteins unfold. And if those proteins denature, that causes holes in the membrane, and the cell will die as a result. And so this is why when we make our slides in lab, we do our heat fix step. Because passing our slide through the heat, through our flame, remember, is going to cause our proteins to denature and coagulate, which then allows the bacteria to stick to the slide, which also kills the bacteria in the process. And so that's why we do our heat fix step. The optimum growth temperature is just like its name suggests. So this is an intermediate between the minimum and the maximum that promotes the fastest rate of growth and metabolism. And so again, like its name suggests, it is the optimal temperature to grow the organism. And these numbers vary depending on the organism, and that's what we're going to look at now. So now we're going to look at the maximum, the minimum, and the optimum temperature to grow different types of organisms. And so what you need to realize is that this chart is showing you a typical psychrophile, for example. A typical psychrophile, the minimum temperature that it can grow is going to be here, so it's the blue, so -20. The maximum temperature that it can grow is about 15. And the optimal is typically between 0 and 10, so typically at refrigeration temperature, so about 4 degrees. So in a minute, we're going to walk through what each of those names means, but psychro refers to cold, phile is lover. So a psychrophile is a cold lover. It grows best at cooler temperatures. And again, it grows optimally at refrigeration temperature. If we call an organism a psychrotroph, psychro refers to cold, troph is eater, so it's a cold eater. It can grow at refrigeration temperature. So notice that it grows down at about 5, which is right around refrigeration temperature. But it doesn't grow well once you get to about human body. So typically it might grow a little bit at human body temperature, but not much. Optimal growth is going to be seen around, it's going to be seen around room temperature, so which is about 25 degrees Celsius. If we look at a mesophile, meso means moderate, phile lover. They love moderate temperatures. Typically, they grow best around human body temperature. So notice that the peak is going to be between about 35 and 45-ish. The minimum goes down to about 10. The maximum goes to about 50. But again, these are the organisms that are typically going to grow best at human body temperature. If we call an organism a thermophile, therm, heat, phile, lover. These are our heat lovers. They are going to grow at a minimum temperature of about 45. So again, notice they don't really grow at human body temperature. They grow up to about 80 degrees. Optimally between about 65 and 75 would be the optimal temperature. If we look at extreme thermophiles, extreme thermophiles grow at extreme high temperatures. So notice they don't start growing till we get at about 65 degrees up to above 130 degrees Celsius, so above boiling. You don't need to memorize exact numbers for these ranges, but you should have an idea and an understanding as to where these organisms would grow, meaning that a mesophile is likely going to be found in the human body. It grows optimally at the human body temperature. Psychrophiles don't grow at all at human body temperature, right? They don't really grow at room temperature, but they do grow optimally around refrigeration temperature. Our psychrophiles are going to be around room temperature optimally, somewhat at human body, and also somewhat at refrigeration temperatures. So they're kind of in between. So we're going to go through for each one, and we're going to talk about where these organisms will be found. So a psychrophile, again, the name means cold lover. So these are organisms that love the cold, and they grow typically between 0 and 20 degrees Celsius, and optimally below 15 degrees Celsius. So usually they grow between 0 and 20, but optimally it's not around 20. Optimally would be below 15. So right around there. For these organisms, storage at refrigeration temperature actually causes them to grow rather than inhibiting them. So in their case, they're going to grow at refrigeration temperature. Natural habitats of psychrophilic bacteria, fungi, and algae would be lakes, rivers, snowfields, polar ice, and the deep ocean. So places where the temperature is cold. These organisms are rarely pathogenic, and the reason for that is that they don't grow at human body temperature. So they're not likely to really cause disease. Next, we have our psychrotrophs, and the psychrotroph, the name means cold eater, right? So phile was lover, troph is eater. So the name means cold eater. They grow typically between 0 and 30, and optimally between 15 to 30 degrees Celsius. Most of these organisms don't cause disease because they don't grow well at 37 degrees, meaning human body temperature. So there are some that can cause disease, but many don't. However, psychrotrophs are usually food spoilage organisms. They're usually organisms that will cause food to spoil. Now, an example of a pathogenic psychrotroph
would be Listeria monocytogenes. So Listeria monocytogenes causes listeriosis. And so in patients who are, or in individuals that are healthy, if you happen to ingest Listeria monocytogenes, it can be asymptomatic, or it might cause food poisoning in the form of diarrhea and vomiting. However, for two groups of patients are most at risk for listeriosis. The first would be immunocompromised, and the second group that's most at risk is the fetus. So for immunocompromised, if they ingest Listeria and it gets into the blood, it causes sepsis. It causes bloodborne infection. If it invades the central nervous system in patients who are immunocompromised, it can cause meningitis. And if it causes meningitis, it has about a 30% mortality rate. So again, when we talk about immunocompromised, we're talking about patients who have undergone chemotherapy, for example, patients who have had an organ transplant and are on anti-rejection medications, patients who have late-stage HIV or AIDS. That would also be immunocompromised because their body is fighting off the virus and their T cells have been compromised as a result. So people that are at risk for listeriosis would be immunocompromised. And then again, the second group that's most at risk would be the fetus. And so for females who are pregnant, pregnant females are often advised not to drink unpasteurized milk or to eat unpasteurized cheese. They're also advised not to eat deli meat that has not been thoroughly heated, because the more common ways that people get listeriosis is through meats and through milk products. And that's because cows also get listeriosis. And so as a result, cow's milk and therefore cheese can have listeria bacteria in it. And so if it's not pasteurized, that bacteria might be present. And if a female is pregnant and she happens to ingest this bacteria and she gets listeriosis, for the fetus, it can cause the baby to be stillborn or to contract meningitis, which leads to brain injury and death and has about a 60% mortality rate. And so again, oftentimes pregnant females are advised, don't drink unpasteurized milk, don't eat unpasteurized cheese, and don't eat deli meats that haven't been thoroughly cooked. And what that means is typically they'll advise you to heat that deli meat until it's steaming. That should kill the listeriosis and therefore make it safe to consume those deli meats. Now, does that mean that's the only food that is the cause of listeriosis? And the answer is no. In recent years, we've seen many examples of produce that's been contaminated with listeriosis. We've seen spinach, we've seen alfalfa sprouts, we've seen lettuce. And so even if, let's say, you are vegan and you don't eat animal-based products, that does not mean that you're safe from listeriosis. It just means that your risk is probably lower, but you could still contract it from other types of food products as well. It's a foodborne illness. And so that would be an example of a pathogenic psychotroph. Now, along those lines and food spoilage and food safety, the way that we store our food is extremely important. And that is that at high temperatures, so if we look at degrees Celsius, so at boiling temperatures, which is 100 to about 65, that temperature, those high temperatures, will destroy most microbes. Now, not all. Some bacteria, for example, endospores, are resistant to high temperatures, but most bacteria will be killed by those high temperatures. However, once you get to about 60 degrees, you're going to get slow bacterial growth, and notice the danger zone, is going to be between about 50 and 15-ish. If bacteria stay for a long period of time at the temperatures within the danger zone, that's going to cause bacteria to multiply very rapidly. And so in that case, typically a good rule of thumb for food safety is if it's left out for more than 2 hours, throw it out. More than 2 hours typically is when you would want to throw it out, because if that food has been sitting for 2 hours, right, and it's been warm, meaning it was cooked and now it's sitting and it's been warm, it's going to sit within that danger zone for a long period of time. And the bacteria will grow rapidly during that danger zone, and some can produce toxins. If you now consume that food that's been sitting out, it's either going to contain the bacteria or contain the toxins that will cause you to become sick. And so an example of this that we've talked about was we talked about B. cereus, right? Bacillus cereus causes food poisoning in rice, because if we cook the rice, right, and let's say that that rice has B. cereus on it, the rice may contain B. cereus endospores. We cook the rice, but endospores are able to survive those high temperatures. If now the food is left warming, if it's left on a food warmer to keep the rice warm but not continue cooking it, the problem with that is that now we've created the perfect environment. There's starch in the rice, so there's a food source. It's moist because we added water to the rice when we cooked it. And it's at a good temperature, which is going to encourage microbial growth. And so B. cereus in rice is going to start to grow and multiply and produce a toxin, so that if you eat that food, if you eat that rice that's been contaminated, typically within 2 to 6 hours after ingestion is when you're going to start to see symptoms of B. cereus food poisoning. If we look at temperatures below the danger zone, if we look at about 15 to about 5, many bacteria survive. Some might grow at that temperature. Refrigeration temperature may allow slow growth. And typically once we get below freezing, so below 0, typically we're going to get no significant growth. So freezing food, for example, is a good way to preserve it, because most microbes can't grow below 0 degrees Celsius. On the right, what you're looking at is that the container that you store food in when you go to put it in the refrigerator, the type of container that you use needs to be well thought out. And what I mean by that is, if you look at the red line, notice the container that the food is put in for the red line. It's a very deep container, much deeper than, let's say, the one in the blue. The blue is much shallower. The problem with that is if you use a shallower container, that's going to be better. And the reason is that you have more surface area to volume ratio, right? You have more surface area in contact with the colder temperature. On the right, when you have a big container and there's very little surface area being exposed to the cold relative to its volume, that means that for the one in the red, the food in the middle, it's going to take longer for that food to cool down, because it's going to take more time for that temperature to diffuse and to cool down that food. And so if you look at the shallower one, the blue container, notice that if you put that in the refrigerator, notice that it gets out of the danger zone fairly quickly. It gets out of the danger zone in about an hour and a half. On the flip side, if you use a container that is really tall, right, and so it's not shallow, but instead it's really big, notice that the food is going to stay in the danger zone for a much longer time, greater than 8 hours. That's going to cause a lot of problems, because in this danger zone, that B. cereus is going to grow and multiply, and it's going to cause food spoilage and then possibly food poisoning. And so you always want to keep that in mind when you're storing food in the refrigerator. You don't want to take like a big pot of soup and put the whole pot in the refrigerator. Instead, it's better to split it into several smaller containers. That's going to help preserve the food better, because if you have a greater surface area to volume ratio, meaning it's stored in smaller amounts, it's going to cool down faster than if you leave it in a big container. So you want to keep that in mind when you're storing your food. So next we have our mesophile, and the name means moderate lever. These are our moderate levers, and these grow typically between 10 and 50, but optimally between 20 to 45. So at room temperature, at human body temperature, and so as a result, these are the most common spoilage and disease-causing organisms. So food spoilage alone, that would be your psychrotrophs or your psychrophiles, because they grow at cold temperatures. But in terms of food spoilage that's going to cause disease, this is going to be much more likely for the mesophiles. And they will cause disease because they grow best, or they grow well, at 37 degrees, which again is human body temperature. So these are the most common pathogenic organisms. So most pathogenic organisms would fall under being a mesophile. And some examples of human pathogens, we have E. coli. E. coli causes sepsis, urinary tract infections, food poisoning in the case of O157H7. Staph aureus would also be a mesophile. It causes skin infections. It causes sepsis. It causes pneumonia. It actually kills more people than AIDS. So Staph aureus is a big problem. Staph aureus also causes foodborne illness as well. So it causes a variety of diseases, but it's a mesophile. And so you have to think about why our body, in response to an infection, why our body would produce a fever. And the answer is that a fever is useful to our body because it increases body temp and mesophiles suffer. So at about 39 degrees Celsius. And so for the mesophiles, they grow best at human body temperature. And in many cases, many of them will slow down growth if it gets above human body temperature. And so a fever is our body's way to try and inhibit microbial growth. And so many microbes that cause disease would be mesophiles. And so our body produces a fever to try and inhibit their growth. Now, in terms of where these organisms would be found in the environment, so in environment, they inhabit animals and plants, as well as soil and water in temperate, subtropical, and tropical regions. It's not going to let me write there, so we'll write it over here, regions. So in the environment, it'll inhabit animals and plants, as well as soil and water in temperate, subtropical, and tropical regions. So basically where the temperature is warmer. And so again, mesophiles are going to be the majority of medically significant microbes. Now, when we call an organism thermoduric, what that means is that they're normally mesophiles. So normally they survive best around human body temperature, but these organisms can survive short exposure to high temperatures. So they can tolerate higher heat better than a typical mesophile. And so these are common contaminants of heated or pasteurized foods. So meaning, again, pasteurization is using low heat to reduce the number of spoilage microbes. And so thermoduric organisms are going to be resistant to that. And so again, these organisms would be common contaminants of heated or pasteurized foods. Examples of thermoduric microbes would include heat-resistant endospore-forming bacteria such as Bacillus and Clostridium, right? They're able to tolerate higher than normal temperatures because of their ability to produce an endospore. Because they can produce that tough keratin shell around the DNA, that is going to make them resistant to heat. And so those organisms would fall under being thermoduric. If we call an organism a thermophile, name means heat lover, right? Thermos, heat, phile is lover. These grow between typically 45 and 80, and optimally, usually between 50 to 60. For these organisms, these live in sunlit soil and water that's associated with volcanic activity, compost piles, hot springs, and other places that are directly in contact with the sun because they like these higher temperatures. Most eukaryotic forms cannot survive above 60 degrees Celsius. So this is unique to usually bacteria. Again, if we call an organism an extreme thermophile, they grow between 80 to 121 degrees Celsius. So they might be able to tolerate the autoclave, for example, which we'll talk more about later. Now, in addition to physical conditions being temperature, pH also affects microbial growth. And we saw this in lab when we looked at the tubes, the demo that was set up in the back, where we inoculated different bacteria under various pH environments. And we looked at what pH do various bacteria grow. Most bacteria grow between a pH of about 6.5 to 7.5, meaning most bacteria prefer a neutral pH. Molds and yeast, so fungi on average, grow best between a pH of 5 and 6. So fungi typically like a slightly lower pH. And you're going to see that when we look at our fungal growths. You'll notice that we culture fungi often on a type of agar called Sabarod's dextrose agar. In that agar, the pH is typically lower, and the purpose of that is to help encourage fungal growth, but inhibit bacterial growth. And so most bacteria, though, are grow best around a neutral pH. So if we call an organism an acidophile, acidophiles grow best in an acidic environment, right? They grow best at a low pH. This is likely to be the case for most food spoilage microbes. And so an example that we looked at for an acidophile, we saw in lab lactobacillus, okay, which is used to make yogurt. We have other organisms. There's one called thermoplasma. It lives in coal piles at a pH of about 1 or 2. There are several types of organisms that can live in extreme acidic environments, but if they're an acidophile, they grow in an acidic environment. So again, low pH. Neutrophils grow best at a neutral pH. And so this is likely to be the case for most normal microbiota or normal flora. So many bacteria in the body that are part of normal flora would fall under being a neutrophile. Right? So E. coli, for example, so on and so forth. But you might notice, so they grow best at a neutral pH around 7. And again, you might recall that whenever we did our experiments with pH indicators, you'll notice that most of the time those tubes started out at the neutral pH. So when we use fennel red, for example, fennel red was used in, let's say, a fennel red broth or a TSI. You might recall that fennel red is orange when neutral. When those tubes started out with the fennel red, they were orange to begin. When we used our bromothiamyl blue, remember that green is neutral. Our OF test started green. Our citrate media started green. They both started at a neutral pH. If we looked at bromocresol purple, started out that weird kind of orange-brown color, which is neutral. So most microbes would fall under being a neutrophile. They prefer a neutral pH, and that's why our media typically starts out at the neutral color when we use a pH indicator. And then we have our alkalophiles. And so our alkalophiles are organisms that grow in alkaline conditions best. Proteus would be an example. Proteus, remember, is also urease positive. And so it has the ability to take urea in the urine, for example, and produce ammonia. And that allows it to colonize and infect the urinary tract. So Proteus can cause urinary tract infections because of its ability to produce ammonia. Now, thinking about what is normally in media that keeps the pH constant. And so in lab, we've talked about this several times. What do we normally put in growth media that's used to keep the pH constant? And the answer, you probably know by now, buffers. So we have buffers in the media that are used to keep the pH constant. So dipotassium phosphate, for example. There are many, many different types of buffers that we use, but usually they're in the media to keep the pH constant. Now, in terms of pH having an effect, so does osmotic pressure. And again, we saw this in the demo in lab. And so if we put a cell in a hypertonic solution is going to have a higher than normal salt concentration. When the salt concentration, so over here, when the salt concentration is higher on the outside, that means that there's more free water in the cell, and the water is going to move from its high concentration in the cell to the low concentration out. And so if you put a cell in a hypertonic solution, the water is going to come out. And in the case of bacteria, it's going to do what's called plasmolysis. The cell membrane is going to collapse in. There's still a cell wall there, but the cell membrane will collapse in, and that will inhibit microbial growth. And so if you think about it, if you think about fruit, for example, if you look at like canned fruit is often stored in a high sugar environment. And you might think, well, wouldn't microbes like that sugar? Wouldn't they want to use that as a food source? And the answer is yes, they would like that. However, if the sugar concentration is too high, that creates a hypertonic solution. And if microbes are in there, it's going to cause the water to come out of the microbe, and it's going to inhibit that microbe's growth. Meats, for example, are often preserved using salt. Same thing. Salt creates a hypertonic solution, and that then causes water to come out of the microbes. So any microbes that might be present are inhibited by the high solute concentration. So osmophiles, osmo refers to osmotic pressure, philes are levers. Osmophiles live in habitats with high solute concentrations. If we call an organism an extreme or obligate halophile, that means that they actually require high osmotic pressure. So not only do they tolerate it, but they actually require it. They need a high solute concentration in order to grow. Other organisms are facultative halophiles. Just like we can have facultative anaerobes that grow with or without O2, a facultative halophile can grow with higher osmotic pressures, so higher solute concentrations, or without it. We also call these organisms osmotolerant. Osmotolerant means that they tolerate high osmotic pressure. They don't require it, but they can grow in the presence of high solute. And again, when we looked at the demo in lab, we compared E. coli and Staph aureus. Staph aureus is osmotolerant. It's able to tolerate 2 to 15% salt. E. coli could not. E. coli could only grow at the low salt concentrations. But Staph aureus, if you think about its normal habitat being the skin, it has to be able to tolerate that high salt in order for its habitat to be the skin. And so that would be an example of an osmotolerant organism. So, question for you. Which of the following temperatures would most likely kill a mesophile? So, I want you to pause the video and think about your answer, and then when you're ready, turn it back on. So, which of the following would kill them? 60 degrees Celsius. So remember that lower than normal temperatures, so mesophiles again would grow best around moderate temperature. So room temperature, human body temperature, right? So 25, 37, 45, those would all be optimal temperatures for a mesophile. Minus 50 is outside of their optimal range. So is 0, so is 9. Those are not within the range that mesophiles grow. However, when you go below the minimum temperature, below the minimum is going to inhibit their growth, but not likely kill them. And that's why many microbes, when we want to store them long term, we're going to store them in the freezer. We even store our bacteria in a minus 70 degree freezer. The minus 70 is just going to inhibit their growth, but it's not going to kill them. But the higher temperatures, the 60 degrees, that's going to cause proteins to denature. And when those proteins denature, that is then going to kill the bacteria. Now, if we look at interactions among microbes, we have different terms for different types of interactions. If we talk about a symbiotic relationship, these are organisms that live in a close nutritional relationship. This interaction is required by one or by both members. Now, usually when we think of symbiosis, we think of it being positive for both. But what you need to realize is that a symbiotic relationship is not necessarily positive for both. If it is a positive interaction for both, we call this so positive, positive. An example of this would be mutualism. And when we talk about mutualism, both members benefit from this type of interaction. So examples of this would be many bacteria that are part of normal flora. They're part of normal microbiota. They benefit us by helping us with digestion, etc. We help them by getting them food or giving them shelter. That would be a mutualistic relationship. Both organisms benefit by this interaction. We also can have what's called commensalism. And in commensalism, one partner benefits, so it's positive, and no effect on the other. So the other member of this interaction doesn't benefit from it. It's not harmed by it. It doesn't really affect the other person or the other thing, the other organism. So an example of commensalism would be some types of normal flora bacteria. Some types of normal flora bacteria might be advantageous for the microbe, but they might not do much for the host. So that would be commensalism. One partner benefits and the other member is not affected. It's not harmed, it's not benefiting, it's ambivalent. If we talk about parasitism, so parasitism, the parasite is dependent and benefits, and the host is harmed. So we call this a positive-negative interaction. One organism benefits, the other is harmed, right? And so the parasite would benefit, and the host would be the one that's harmed. Now, if we look at non-symbiotic relationships, the organisms are free-living, and the relationships between them are not required for survival, meaning they can survive on their own. However, it could be beneficial or it could be a negative interaction. So the first one is going to be synergism. And so in synergy, the members cooperate and they share nutrients. And so an example of this that we're going to talk about in a minute is going to be a biofilm. And so we'll come back to that and talk about that in a minute. So organisms that live within a biofilm, remember that a biofilm is an intermicrobial community living in slime. So these organisms can cooperate, they can share nutrients, etc. On the flip side, antagonism. In antagonism, some members are inhibited or destroyed by others. So competing for a similar resource would be an example of antagonism. And so if two organisms are both competing for some limited food source, for example, that would be a type of antagonism. It's not a positive interaction. It's actually negative. And these two members are competing for some common resource. So that would be antagonism. So if we look at a biofilm, again, this is the epitome of synergy. This is a good example of synergy. And again, a biofilm is going to be a mixed community of bacteria and other microbes that are attached to a surface and to each other. So again, it's a complex intermicrobial community living in slime. And so again, the way that this biofilm is going to form is that some pioneer bacteria is going to attach and colonize the surface. So an example could be on teeth, for example. Strep mutans can bind to the surface of the teeth, and that could be your pioneer. Other microbes, so the pioneers secrete some extracellular material that helps them to attach to the surface and also allows other microbes to attach. Those initial colonizers produce chemicals, and they do, remember, quorum sensing. And they're going to quorum sense, and they're going to send out a signal that's going to attract other bacteria to where the biofilm is. Now, other microbes are going to join in on the biofilm. They may add more slime, but they might not. But these other organisms are going to be living in slime, and the attached cells are stimulated to release chemicals as the cell population grows. And so the interaction between these bacteria is an advantage, right? It's not necessarily required. The organisms can survive on their own. However, it does give them a benefit. So next we're going to look at culturing microbes. And so some of these next slides I'm going to go through very quickly because we did talk about them in lab, but I still want to cover them again. And so when we talk about media and we talk about agar, remember that agar is a complex polysaccharide derived from seaweed, and agar is used as a solidifying agent for culture media in Petri plates, slants, and agar deeps. And remember that the purpose of agar is generally not a food source. It's not metabolized by the microbe as food. Students often see polysaccharide and they think sugar, and they say, well, it's a sugar, it must be used for food. It's not. It's a complex polysaccharide that the microbes can't degrade and they can't use as a food source. And think about why you know that to be true as well. Because if bacteria were using it as a food source, right, and they were hydrolyzing or breaking down that polysaccharide, that agar plate would go from being a solid to a liquid. Think of gelatin, for example, right? If the organism has gelatinase, they can hydrolyze the gelatin and it liquefies the gelatin to produce the polypeptides and amino acids. You would expect that if agar is a solidifying agent and it's used as a food source, that would mean that the agar would go from being a solid to a liquid, and we don't see that. So that tells you right there, the fact that our plates don't get liquefied tells us that the agar is not there as a food source. It's simply there to act as a solidifying agent. Again, the way that agar is made is that you take that powder, you mix it with water, you heat it above 100 degrees. That is going to cause the agar to go into solution just like Jell-O. It's going to liquefy at 100 degrees. Then when you pour it into plates and you let it sit at the desk, for example, it's going to solidify and become a gelatin at about 40 degrees Celsius. And so it's going to solidify and it's going to give us that gelatin that we're used to working with. Now, chemically defined media, again, is media where the exact chemical composition is known. There's no peptones in the media, there's no beef extract, no yeast extract, no ingredients that's going to vary from batch to batch. We know exactly what's in the media. This is used to culture fastidious organisms, so organisms that may require specific growth factors, so they have very specific nutritional needs. Those might be grown using chemically defined media. Chemically defined media is also used for certain assays. And so if you think about, we talked about a very specific example of a chemically defined media. So see if you can think about a type of chemically defined media that we've used in this class. And so if you said citrate, right, our Simmons citrate agar, that would be an example of a chemically defined media. Because in that media, you might recall there were no peptones, there was no pancreatic digestication, nothing like that. In that media, the only carbon source we gave the bacteria, the only carbon source was the citrate. The citrate was the sole carbon source. And so remember, in that test, a true negative has no growth, because if bacteria can't use the citrate as their sole carbon source, they can't grow. But that's not the case for many of our other tests, like a fennel red broth, for example. It has peptones in it, and even in the negative bacteria should still grow because it should be able to use something as a food source. In chemically defined media, right, if the bacteria can't use the citrate, they can't grow. And so that would give you your negative results. Complex or undefined is another name that's used interchangeably. Complex or undefined media contains extracts and digests of yeast, meat, or plants, and the chemical composition can vary from batch to batch. So examples of complex media: nutrient broth, nutrient agar. These would be examples of complex media. So if I look at the ingredients in nutrient agar, we have peptones. Peptones are partially digested proteins. Sometimes you'll see that in the media as pancreatic digestication or pancreatic digest of gelatin. Basically, it's partially digested proteins, and it's there as food for general growth. It varies from batch to batch, which is why we call that complex media. You can also see in this media beef extract. That also makes it complex. It varies from batch to batch. We have sodium chloride. Sodium chloride is, remember, there for salt balance. Agar is there as the solidifying agent. And then we have to dissolve the agar in water. And so complex media contains ingredients that vary from batch to batch. Now, reducing media is used for the cultivation or the culture of anaerobic bacteria. And this contains chemicals. For example, a thiobroth contains sodium thioglycolate that takes O2 and it reduces O2 into water. So we had sodium thioglycolate, we had L-cysteine. Both of those ingredients reduce O2 to water, and that makes anaerobic. And so our thiobroth would be a reducing media. Also our anaerobic jar. If you recall our anaerobic jar, in the jar we placed that envelope, and that envelope had our anaergen chemicals in it. And those anaergen chemicals were there to reduce the O2 to water, which made our chamber anaerobic. And therefore it allowed us to culture those anaerobic bacteria. So we could use reducing media, we could use the anaerobic jar. In some cases, we actually use anaerobic chambers, which is what you're seeing on the right here. And so that environment in there that you're working with is going to be anaerobic, and it allows you to culture anaerobic bacteria. So when working with bacteria and other types of microbes, different microbes can be given different biosafety levels. And depending on an organism's biosafety level, that will dictate what types of precautions must be used when handling those organisms. So the lowest risk microbes fall into the category of biosafety level 1. And so you can see down here in yellow, biosafety level 1 is the lowest risk microbes. They're not known to consistently cause disease in healthy adult humans, and they have minimal minimal known risk. So examples of BSL-1s would be like Saccharomyces cerevisiae, which is baker's or brewer's yeast, lab strains of E. coli that we continuously use throughout the class, as well as other non-infectious bacteria. And so for BSL-1 type organisms, basically you need to use protective personal equipment, so PPEs. That includes lab coats, right? You guys all wear lab coats in the class. In certain instances, it might include gloves or even goggles, for example. In a BSL-1, people who are in a BSL-1 need to practice hand washing before and after the lab, decontamination of surfaces, avoid aerosols, all the different things that we currently do in our class. BSL-2, if an organism is put into BSL-2, this is moderate potential hazard to personnel and the environment. This includes bacteria and viruses that cause mild disease to humans or are difficult to contract via aerosols. So an example of a BSL-2 and one that probably surprises people is HIV. HIV is classified as BSL-2 because it's not easy to contract via aerosolize, right? It has to get into the blood. And so HIV would fall under BSL-2. Staph aureus, which we use in the lab, falls under BSL-2. Hepatitis A, strep pyogenes, which we will also work with in the class. Borrelia burgdorferi, which causes Lyme disease. Salmonella species, etc. And so for BSL-2 labs, the safety would include, again, all of the things that are in biosafety level 1. So personal protective equipment, so gloves, lab coats, goggles if needed. The lab would need to have an eye wash station in addition to washing your hands. If the bacteria is a risk for aerosolizing, oftentimes in a BSL-2 lab, you'll also use a biosafety cabinet or what we call a hood to work with bacteria. You'll see that in a minute. In terms of biosafety level 3, biosafety level 3 are microbes that are either indigenous or exotic, and they can cause serious or potentially lethal disease through respiratory transmission. So examples of this would be Yersinia pestis, which causes the plague, Mycobacterium tuberculosis, which causes TB, SARS. Remember that SARS is a type of coronavirus. We're all dealing right now with COVID-19, which is also a type of coronavirus. COVID-19 would fall under BSL-3, so that would be an example of a BSL-3 type organism. Rabies virus, West Nile virus, hantavirus, these would all fall under being BSL-3. And so these have very serious and potentially lethal diseases, often through inhalation. This has strict control access to the lab, so you must have clearance in order to even get into the lab. If immunization is available, people who work in those labs should have immunization against the organisms. Depending on the organisms, people who work in these labs might use a respirator, certain types of gowns, not using recirculated air, etc. would be used for biosafety level 3. Biosafety level 4 is used for dangerous and exotic, posing a high risk of aerosol transmitted infections. Infections caused by these microbes are frequently fatal and are without treatment or vaccines, meaning if you happen to contract this, there's nothing we can do to really treat the disease, and there's no vaccine to prevent the disease. So examples of this would be Ebola, right? And so we've seen outbreaks of Ebola. Smallpox, there's only 2 labs in the world that still have the smallpox virus in them. And so that is very important to keep that contained. It would be a biosafety level 4. And so in a biosafety level 4, You'll have to change your clothes before entering. You have to shower upon exit, decontaminate all materials before exiting. People who work in these labs wear full body air supplied positive pressure suits, etc. And so we will look at some of these types of protective equipment in just a minute. So this is a slide from the CDC, and it basically is comparing the different biosafety levels and what types of equipment would need to be in these labs to work with these types of organisms. So in a biosafety level 1 or BSL 1, you would have controlled access. So you would have to have a door that can lock. You need to have a sink for hand washing. There needs to be a sharps hazard. So that's going to be a sheet showing about what to do. Personal protective equipment. So again, lab coat, gloves, potentially goggles, depending on what you're working with. A laboratory bench, and an optional would be an autoclave. We often use an autoclave to decontaminate our materials. That would normally go in our red biohazard bag. For a biosafety level 2, it builds on a lot of the things that we already talked about. So a sealed door, a sink, a sharps hazard policy, personal protective equipment, a lab bench. But now we have number 4, which is going to be our physical containment device, which is a biosafety cabinet. And so this biosafety cabinet contains a glass that goes up and down, and it allows you to work within what we call a hood, which has airflow that protects people from the microbes coming out at the person working with it. So that would be a biosafety level cabinet, and then also an autoclave. Now, biosafety level 3, we start to get more intense in terms of the requirements. So in a biosafety level 3, it has a self-closing double door access. So notice that there's 2 doors leading into this, and in between there is a shower for exiting the room. So here's the shower there. We still have to have a sink. We still have a sharps hazard warning policy, sealed penetration, so basically preventing things from leaving the lab. In some cases, for certain BSL 3s, respirators are worn as protective gear. We still have our biosafety cabinet, we still have our lab bench, we have our autoclave, but now we really start to see exhaust HEPA filters. So again, the air that leaves the room is not going to be recirculated. It's going to be filtered through this HEPA filter, and also a decontamination system. In terms of BSL 4, BSL 4 is the most strict out of all of the different types of biosafety. And so if you look at what's required in a BSL 4 lab, again, self-closing double doors, controlled access, sharp hazards, hand washing sinks, sealed penetrations, physical containment surface, so this biosafety cabinet, positive pressure protective suit. So again, this full sealing suit that you see here. A laboratory bench, an autoclave. It has a chemical shower. It has a personal shower out. Again, it also has supply and exhaust HEPA filters, as well as this effluent decontamination system. And so I put on this video, or on this slide, a link that you can go to and watch. And what you can see on this video is basically they take you on a tour in a biosafety level 4 lab. So if you're curious and you would like to see all the different types of precautions that go into working in a biosafety level 4 lab, that's a cool video to go check that out. In a minute, I'm going to show you 2 quick videos about how to enter a BSL 3 lab, as well as how to exit a BSL 3 lab, so that you can see the level of effort that has to go into preventing contamination. So to continue on about types of media, the next two types of media are media that you're going to see a lot in the end of the semester when talking about lab. And so the last two main types of media that we'll talk about is selective media. And selective media is like the name suggests, it selects for the growth of some microbes while inhibiting the growth of others. And so it contains inhibitors that's used to suppress growth of some group of microbes. Differential media is a little bit different in that it allows us to differentiate or to distinguish colonies of different types of microbes based on differences in species-specific metabolism. So it allows us to distinguish closely related bacteria based on the types of metabolism that they do. Some media have both selective and differential ingredients. So if we were to look at MSA, MSA stands for mannitol salt agar. And again, we're going to talk about this a lot in lab. But mannitol salt agar is used to presumptively identify Staph aureus. Now, presumptive means that if we get a positive on this test, we can say with about 95% confidence that in fact the bacteria is Staph aureus. And so this is a special type of agar that has ingredients that allows us to identify Staph aureus. So when we look at the ingredients that are in the mannitol salt agar, the selective ingredient, the ingredient that's going to select for the growth of some organisms but inhibiting others, the selective ingredient is going to be salt. So sodium chloride. And it has a salt concentration of about 7.5%. So this high salt concentration selects for osmotolerant organisms. So what that means is that on this plate, the salt concentration is high, and so organisms that are not osmotolerant are not going to grow. And so if we look at this plate, you can see this uninoculated. It actually would look the same in that quadrant if we had inoculated with E. coli. E. coli is not osmotolerant, and therefore if inoculated on an MSA plate, it's not going to grow because it's not osmotolerant. And so sodium chloride in this case would be our selective ingredient. It selects for the growth of some while inhibiting others. Bacteria found on the skin, on the other hand, would be considered osmotolerant because, again, bacteria found on the skin would likely be able to tolerate high salt. So Staph aureus could grow on this plate, Micrococcus luteus, which is another type of skin bacteria, Staphylococcus epidermidis, another type of skin bacteria. Those types of organisms could all grow on an MSA plate. So now, if we need to distinguish Staph aureus from, let's say, Staph epidermidis, now we need a differential ingredient. And the differential ingredient is going to be the ingredient that allows us to differentiate based on metabolism. And so in this case, our differential ingredient is mannitol. And mannitol is a sugar that Staph aureus ferments, but Staph epidermidis does not. So both Staph aureus and Staph epidermidis would both grow on the plate because they're both osmotolerant, but we need a way to distinguish between whether the growth that we're seeing is Staph aureus or Staph epidermidis. And so our differential ingredient is going to be our mannitol. And so again, mannitol is a sugar, Staph aureus can ferment it, and Staph epidermidis cannot. Now, also included in this media is going to be our phenyl red. Phenyl red, remember, is a pH indicator. And so based on what you know about phenyl red, what color is it when it's acidic? And the answer is yellow. And remember that that's true for all of our pH indicators except one. And remember that the only exception to that rule for the ones we use in our class would be the methyl red. Methyl red is red when acidic. Remember, it's the opposite of phenyl red. Everything else, bromothiamyl blue, acidic is yellow. Bromocresol purple, yellow is acidic. Phenyl red, yellow is acidic. Yellow is always acidic. So the yellow is going to be acidic, and the pink, the magenta, oops, give that a little more space. Pink is going to be alkaline. So notice that where Staph aureus is growing is yellow. Does that make sense to you conceptually? And it should, because I told you that Staph aureus ferments the sugars. Remember that when organisms ferment sugar, the pH of the products is going to produce acids. And so because Staph aureus is able to ferment the sugar, it's going to produce acids, which lowers the pH, and it causes the phenyl red to turn yellow. On the flip side, Staph epidermidis, Staphylococcus epidermidis, does not ferment the mannitol, so it does not produce acids. So what would you think it uses as a food source instead if it doesn't use the mannitol? And based on everything we've learned so far, hopefully you got to the answer being peptones. So the Staph epidermidis use the peptones for metabolism. And remember that when an organism uses peptones for metabolism, it's going to produce alkaline products. Those alkaline products are going to raise the pH, and it's going to cause the phenyl red to turn pink. And so what you're seeing on this MSA plate is that Staph epidermidis will turn the agar yellow, Staph epidermidis will turn it pink. And if I were to inoculate this with E. coli, for example, which is not osmotolerant, it would not grow and it would look like the uninoculated. And so MSA plates can be used, and you guys will learn about this for lab, that MSA plates can be used for nasal swabs. And if we were to do this in class, you could test your nasal swab and you could see if you were a carrier of Staph aureus. And so you would take your nasal swab and you would swab it on an MSA plate, and you would come back 48 hours and look at your plate. If the colonies that grew turned the agar yellow, then you would have presumptive positive for Staph aureus. If, on the other hand, the plate was pink, the colonies on the plate were pink, it's other type of skin bacteria that is not Staph aureus. So again, Staph epidermidis, it could be Micrococcus luteus, it could be other types of skin bacteria, but it's not Staph aureus. And so this would be our selective and our differential media. And so in lab, you're going to learn a lot about different types of selective and differential media. So this slide is just summarizing the different types of media. So again, chemically defined media is used for the growth of fastidious organisms. So most chemoautotrophs or photoautotrophs. We know the growth factors that the bacteria requires, and we give them to them for growth. Chemically defined media is also used for microbiological assays. So again, like the citrate test. Complex or undefined media is used for the growth of most chemoheterotrophic organisms. It contains a variety of different food sources in there, so peptones, beef extract, yeast extract, etc. If we talk about reducing media is used for the growth of obligate anaerobes. So it has ingredients like sodium thioglycolate or L-cysteine, which is used to make the media anaerobic. Selective media is going to suppress unwanted microbes but encourage desired microbes. So it's going to select against certain organisms and select for the growth of others. Differential ingredients or differential medias are used to differentiate colonies of desired microbes from each other, usually by differences in metabolism. So meaning some types of organisms might be able to utilize that food source and others don't. And again, you're going to see that a lot more coming up in lab. Enrichment media is similar to selective media, but in this case, it's actually designed to increase the number of desired microbes to a detectable level. There's an E. coli enrichment media that can be used to help enrich the population for E. coli. And so these are just the various types of culture media that you will see. So the last part of this lecture is going to look at reproduction in prokaryotic cells. And when we talk about reproduction, we are talking about an increase in the number of cells and not an increase in cell size. And so that's something to be careful with, because oftentimes students will think that it's an increase in the size of the cell. That's not true. It's an increase in the number of cells, meaning that the population is going to get larger. And so in the case of reproduction in prokaryotes, this is always going to be asexual. No partner required. Offspring are identical to parental cell. So asexual reproduction results in offspring that are identical to the parent cell. Now, there is a caveat to that, and that is that in some cases, when a cell goes to reproduce, occasionally there is a mutation, which is a change in a DNA sequence, which could lead to slight changes between the parent and the offspring. But that's different than sexual reproduction, where two partners come together and you get a lot more genetic diversity. Asexual reproduction generally results in offspring that are identical to the parent, again, barring any mutations. So in terms of types of reproduction in prokaryotic cells, one type is binary fission. And binary fission is where bacteria is going to divide into two, and you end up with two genetically identical, equally sized cells. If we look at budding, budding is a type of reproduction where a small outgrowth comes off of the main cell. And so originally when that cell divides into two, the new cell that's forming starts out smaller and then will gradually increase in size. Yeast also do budding. It's still an asexual reproduction, but it doesn't equally divide the cell into two equal sizes from the beginning. One cell starts out smaller and then will gradually increase in size. We can also have what are called kinidiospores. Kinidiospores are formed in a type of prokaryotic cell, Actinomycetes. This is a type of bacteria that is fungal-like. So this is a fungal-like bacteria, and it reproduces using kinidiospores, like fungi do as well. Some prokaryotic cells reproduce by fragmentation of filaments, etc. But again, this type of reproduction is going to be asexual. The offspring are going to be identical to the parental cell. So if we look at binary fission, in binary fission, one cell divides and becomes two. And so what's going to happen is you have a young cell, and this young cell is actively dividing. And the first thing that's going to happen is that the chromosome is going to be replicated. Now, most prokaryotic cells have one circular chromosome. And so the bacteria is going to replicate that chromosome into two pieces. And those two parts are going to move to opposite sides of the cell. The parental cell is going to elongate. It's going to get bigger. And eventually, it's going to start to pull its cell envelope together in the center of the cell. So it's going to start to pinch in, and a protein band is going to form in the center of the cell. Then that's going to form a septum. It's going to produce this wall that's going to physically divide these cells into two. And that central septum is going to divide that one cell into two. And then these cells are going to be two identical daughter cells from the parental cell. In some cases, the cells completely separate. In the case of other bacteria, if they are strepto, remember that strepto means that they are in chains. And so you could have streptobacillus, and so those bacilli are arranged in chains. They're still hooked up and linked together. And in other cases, they will completely separate. And so this is just showing you an electron micrograph of a species of bacillus starting to undergo binary fission. And you can see here that there's this partially formed crosswall that's starting to divide the cell into two. And you can see DNA, two DNAs in these nucleoid regions. Remember, they're not contained within a membrane, so a nucleoid region. And they're in opposite sides of the cell. And then eventually that crosswall is going to form. It's going to form a septum, which is going to divide that cell into two. So when we talk about bacterial growth, we talk about bacteria's generation time. And that is the time required for a cell to divide and for its population to double. So basically, how long does it take the cell to divide, meaning to go from one cell to two? Most bacteria have a generation time of about 30 to 60 minutes. That's on average. Some bacteria, the fastest known bacteria, have a generation time of about 9 minutes. So every 9 minutes, that bacteria is able to replicate and go from one cell into two. Some bacteria have much longer generation times. Mycobacterium leprae, which causes leprosy, has a generation time of 10 to 30 days. So it reproduces very, very slowly. Some environmental bacteria have generation times that are measured in months. So it just depends on the bacteria. There's a lot of variation from bacteria to bacteria, but most pathogens generally have a relatively short generation time. So E. coli, for example, has a generation time of about 20 minutes. So in 20 minutes, it goes from one cell to two. Another 20 minutes, it goes from 2 cells to 4. And then 4 cells to 8, 8 cells to 16, 16 to 32, and so on and so forth. And so the number of cells in each generation is 2 to the n power, where n is the number of doublings or generations that have occurred. So if you imagine that a cell has 30 generations, 2 to the 30th power, the population would be 1 billion. So think about that. 30 generations, population would be 1 billion. So if you think about E. coli, and E. coli reproduces every 20 minutes, right? And so every 20 minutes would be 1 generation. So if you're thinking 30 generations, right, that would be 10 hours. So within 10 hours, it would go from one single E. coli to 1 billion in 10 short hours. So that tells you how fast this generation time or how fast this population grows. It grows logarithmically or exponentially, meaning as the population gets larger, it's going to increase the population size at a faster rate. So what you're looking at here is you're looking at different representations for how the cell division can be plotted. And so what you're looking at on the left is showing you if you start with one cell and you get to one generation, that's going to go from one cell to two, right? And so 2 to the 0 for the one cell, 2 to the first, we have 2 cells, right? After 2 generations, we're going to have 4 cells, so 2 to the second power, 4. 3 generations, it's going to go from 4 to 8, 8 to 16, 16 to 32. And so notice that your population is going to grow exponentially. It's going to get bigger and bigger and bigger. Now, if that's plotted on an arithmetic scale, so this is an arithmetic scale, meaning that the units are going to be even. If you plot this growth curve on an arithmetic scale, you get this interesting growth curve, meaning it looks like initially there's no growth, and then all of a sudden the population just takes off, which is kind of a misleading way to represent the data, because in fact, even at these earlier times, the population is still growing. It's growing at an even rate. It's just that your population is starting small. And so when it divides, the numbers are small initially until you get to a point, and when it doubles, it gets exponentially larger. So a better way to graph this would be on a log scale. And so notice that if you plot this on a log scale, this is a better representation because it's showing you that the bacteria is dividing at a constant rate. It's always dividing at a constant rate, right? It's not changing depending on the number of bacteria. The cell division rate is still the same. It's just the population size looks a little bit different if you plot it on the arithmetic versus the exponential scale. And so most often than not, we typically, when we plot growth curves for bacteria, we're going to plot it on a log scale. And that's going to show that increase in the population size. It's not going to make it look like it's just flat to start with. So this is just showing you again, looking at the number of cells and looking at the log scale of that. So after 5 generations, that would be 2 to the 5th power, or 32 cells. After 10 generations, it's going to be at 1,000. After 15, it's going to be at 32,000. So notice that going from the first cell to 5 generations, it only went from 1 to 32. Now for each subsequent generation, so when we get to 15 to 16, notice 32,000 to 65,000. And then 131,000. Notice the numbers are getting large much, much faster. And by the time we get to 20 divisions, we're already at 1 million cells. And so this is just showing you that, again, this growth is exponential. If you plot it on a arithmetic scale, it's going to look like it doesn't grow very much, and then all of a sudden it takes off. And so a better way to show that it's constantly dividing would be to actually plot it on a log scale. So this slide is looking at the 4 phases of growth. And so we're going to put some notes for each of these 4 phases. So in the first phase of growth, is called the lag phase. And the lag phase is going to appear relatively flat. And this happens because newly inoculated cells require a period of adjustment. So the cells are starting to adjust to their new environment, right? So an example of this would be like if we transferred bacteria from a slant into a fennel red broth. Initially, the bacteria would have this lag phase. They would be getting accustomed to their new environment. They would be preparing for cell division, but cells are not yet multiplying at maximum rate. So they're not yet growing at their maximum rate. They're adjusting, they're acclimating to their new environment. And furthermore, because this population is so sparse, meaning you're starting with so few bacteria to begin with, because the bacteria is so sparse or so dilute, oftentimes sampling them or looking at how much bacteria might be there might miss those cells that have divided. And so you're going to see this lag phase, this phase where you're not going to see much growth because they're just starting to acclimate to their environment. They're synthesizing their macromolecules. They're basically getting themselves ready for cell division, but they're not yet quite dividing. The next phase is the exponential growth phase, or the log phase. You'll also hear it called the log phase. So that's the second phase. And so this is where growth curve, or growth of microbes, increases at a constant rate. And so this will continue. So will continue as long as cells have adequate nutrients and environment is favorable. So this is going to be your fastest rate of growth. And so bacteria are going to increase at an exponential rate. And so if you look at your log scale, you're going to see that this population is growing at a steady rate. This is your fastest rate of division until eventually they're going to get to what we call the stationary phase. And at the stationary phase, cell division is slowing down. Birth rate equals death rate. So no change in population size. So no change in population. And so in this case, this is caused by depleted nutrients plus waste products building up. So what that means is that at some point, the organisms that are in that culture are going to run out of resources. Food is going to become scarce, right? Because as the population increases, they're going to be utilizing the food in the culture. So food's going to become scarce. They're going to produce waste products. Those waste products might be acids, for example, if they're metabolizing the sugar. Those acidic products are going to change the environment, which is going to inhibit microbes from growing. And so eventually you're going to get to the stationary phase, and you're going to see no change in population size because your birth rate is equal to your death rate. And then lastly, so number 4 is going to be our death phase. And in our death phase, cells begin to die at an exponential, constant rate. So cells begin to die at a constant exponential rate. And this is going to be due to a buildup of waste products, a lack of nutrients, etc. And the speed with which death occurs is dependent on the resistance of the species, as well as how toxic the conditions are. And so each species is going to have a different rate at which the death phase occurs. And typically this rate at which the bacteria dies is typically going to be slower than the exponential growth phase. Both are going to be at a constant rate, both are exponential, but typically the rate at which the death phase occurs is going to be slower than the rate at which the exponential growth curve occurs. Some cells within the culture may remain viable. They might not be growing. So notice that it didn't go down to nothing. Some cells might remain viable, but not be growing. And it might be missed in colony counts if you're trying to determine how much bacteria is there. So if we now go back and put some additional notes, so for our lag phase, this is high metabolic activity. Cells adapt to new environment, can last for 1 hour to several days, and we get no increase in population size. Next, we have our log phase. This is our fastest rate of division, the fastest rate of division. We have exponential growth, so exponential growth. And at this point, birth rate is greater than the death rate. So our population increases. Then we get to our stationary phase, and at our stationary phase, our birth rate is equal to our death rate. So no change in population size. This occurs because of exhaustion of nutrients, accumulation of waste, and harmful changes in pH. And then our last is going to be our death phase. And in our death phase, our birth rate is less than our death rate. Cells are dying at exponential rate. So now, if you think about this, right? And so in our death phase, cells are dying at an exponential rate. And if you look at this all together, all of these 4 phases, I want you to think about during which of these 4 stages would you expect to see endospores form? So which of these phases would you expect to see endospores form? So if you need to, you can pause to think about it. And then when you're ready, push play and I'll go over the answer with you. So which phase will endospores form? Endospores will form in the stationary phase. And so endospores would form during the stationary phase because this is when bacteria would experience their condition as being harsh. And this, in response to these environmental changes, that would be where the endospores would form. It's not going to form during the log phase when they're still dividing. It's not going to occur in the death phase because by that point it's too late. It's going to occur during the stationary phase because that is when the bacteria would experience their environment as being harsh. Now, the tendency for populations to exhibit phases of rapid growth, slow growth, and death has implications in microbial control, in food microbiology, and also in culture technology. Microbes in the exponential growth phase are more vulnerable to antimicrobial agents and heat, meaning that bacteria is more likely to be able to be killed if they're in their fastest phase of growth. So if they're in their exponential growth curve, they're more susceptible to these antimicrobial agents. Actively growing cells are more vulnerable to conditions that disrupt cell membrane and binary fission. A person actively shedding bacteria in the early and middle stages of an infection is more likely to spread it than a person in the later stages. And so this is why it's useful to know where bacteria are in their growth curve, because early on, those bacteria are more metabolically active than, let's say, bacteria that have been in the body for a while. So there are several methods that can be used to culture and analyze population size. So analyze population size without culturing. And the first method would be to measure turbidity. And remember that in lab, we have used turbidity as a method to determine how much bacteria is grown. And so remember that the more that the microbe has grown in the solution, the more turbid the solution becomes. It becomes a lot more cloudy, right? And when we vortex our tubes, we're looking for that turbidity or that cloudiness. Now, we can use what's called a spectrophotometer. This is our spectrophotometer. And it's a machine that is going to measure the amount of light that passes through the sample. So in the case on the left, that first sample has a low turbidity, meaning there's not much bacteria in the sample. So when light is going to be shown through the sample, a high percentage of the light is going to pass through. You're going to end up with what we call a low absorbance. Very little light was absorbed by the solution. Most of the light passed right through. So when we look at our spectrophotometer, we can either have those numbers represented in transmittance, meaning the higher the transmittance, the lower the population of bacteria, or the lower the absorbance, the lower the amount of bacteria because there's nothing absorbing the light. On the flip side of that is if we have high turbidity. So if we have a lot of turbidity and the light passes, or the light hits where the bacteria is, that light, when it hits the bacteria, is going to cause the light to scatter, and you're going to get a low amount of light transmitted. So if I were to take a note here, so this is a high transmittance or low absorbance. There's two ways that can be represented. If we have high turbidity, that means an increase in population, meaning we have more bacteria. We get low transmittance or high absorbance because the bacteria is causing that light to reflect, and therefore it's not going to pass through the sample. And so this is a way that we can quantitatively determine the relative population size. And so when we did this in lab, we did this very subjective, right? You guys looked at them and you just looked at turbidity and you compared which one had more turbidity than another. But if you really needed to know if bacteria was growing exponentially, using turbidity and using a spectrophotometer, you could quantitatively look at how your population size is changing over time, because the more turbid the solution, the larger the population size is, and that would tell you something about how the bacteria is growing. There are other types of population size determinations. We can have a variety of different counting methods. So one could be a direct cell count, measuring microscopically. We can have a cytometer. So you can see this little slide up here, and it has these ridges, and you would pipette the sample in, and on there is a grid. And you could use the grid to keep track and to count how many microbes are in the sample. The downside to this approach to direct cell counts is that just by looking at it, there's not a good way to distinguish between living cells and dead cells. So that is a drawback of this method. It will tell you how much bacteria is there, yet it won't tell you what percentage of those cells are living or dead. We can also have an automated cell count, so what's called a Coulter counter. And so what happens in a Coulter counter is that the sample in a liquid is passed through a tube, and that tube is very narrow, and the sample's going to pass through, and there's a counting component, an electronic detector, that's going to be able to detect each time a cell passes through. And so it's going to be an automated counter to determine the number of cells in the sample. So that's a Coulter counter. We also can have a flow cytometer. A flow cytometer works very similar to a Coulter counter. You're still going to pass the sample through a narrow tube, but the flow cytometer does several other things. It can measure cell size. It's able to differentiate between living and dead cells. And we will talk more about flow cytometers when we look at our white blood cell counts. And then the other thing that we can do to measure or analyze population size without culturing would be to do genetic probing. So that would be the third method. And so this is going to use what's called real-time PCR to quantitate how much bacteria and other organisms are present in an environmental or a tissue sample. And so this is going to use the amount of RNA present to determine how much bacteria is present. So that would be genetic probing. So this is a class paper. And so what I'm going to do for this is I'll read this here, but I'm actually going to post this class paper on a discussion board in Canvas because we still need points for our participation for lecture. And since we're no longer meeting in lecture, we will do our class papers through the discussion board instead. So I will post this on the discussion board. I'll open it up for a period of time. You'll post your answer. Remember, it's participation. So if you don't get it right, you're not marked against. It's just to see, are you thinking about this and are you analyzing what you've learned in class? So this class paper says nitrogen and phosphorus are added to beaches following an oil spill to encourage the growth of natural oil degrading bacteria. Explain why bacteria do not grow if nitrogen and phosphorus are not added. Hint, think of what oil is composed of too. So when you think about this, you want to think about what are the elements that bacteria need in order to grow. How do they get those elements normally? What molecules use those elements? So meaning like nitrogen, what do bacteria use nitrogen to produce? And so when you're thinking about this, you want to think about what elements is the oil providing to the environment, meaning what elements is the oil going to be made of, and then why do you still need nitrogen and phosphorus? And so you're going to post your answer on Canvas, and then once the discussion board closes, I will post the correct answer.